Hello, welcome to my presentation on the clinical scale production of extracellular vesicles in a hollow fiber bioreactor. I'm John Cadwell. I like to take the Michener approach to cell culture. It's important, I think, for you to understand that cell culture is a relatively new technique in the laboratory. First attempts at culturing cells in vitro or in glass outside of the body was by Harrison in 1906. And he used frog neuronal implants on a plasma clot, a three-dimensional cell culture matrix. Subsequently, we moved from glass into plastic. It wasn't until the late 50s that we developed the HeLa cell line. We required proteolytic enzymes to make for single-cell suspensions. Laminar flow hoods didn't come onto the scene until the late 70s. Scaling up of cell culture has always been about more surface area, but fundamentally, current two-dimensional cell culture is an inherently non-physiologic process. We are growing cells subconfluent on a non-porous two-dimensional plastic dish in the presence of fetal bovine serum, which in itself is non-physiologic. Serum is plasma with the clotting factors removed. The only time that cells will see serum is in an activated state. So it's important to understand that cell culture as practiced today is inherently non-physiologic. All of these techniques, to a greater or lesser degree, will subject the cells to what we call the feast or famine cycle, which means that glucose starts out high, lactate starts out low. There's this period of time that the cells are attaching to the plastic and starting to secrete some of their own cytokines that we call the lag phase. After the lag phase, glucose consumption will increase, lactate production will increase. The cells at their optimum density and homeostatic conditions for only a short period of time for optimum protein production, EV production, and cell physiology. And then just when things are getting good, it's time to trypsinize everything. We have to split the cells. The reason that we split the cells is that nutrients come from the top down, and the bottom layer of cells do not get gas and nutrients, so that they die and peel off and float away. So we must maintain the cells in a subconfluent monolayer culture. This, again, is inherently non-physiologic. Cell culture conditions are constantly changing, and the cells spend a very small amount of time out of the total time in culture in their optimum homeostatic cell culture conditions. Hollow fiber is fundamentally different from any other way of culturing cells, and it is a three-dimensional, more in vivo-like way of growing cells. And it's based on the principle that each one of these little hollow fibers is like a filter. It's shaped like a drinking straw. It's about 200 microns in diameter. We seal them in the ends in this polycarbonate shell using a polyurethane potting material so that anything that goes from the ends of the cartridges necessarily goes to the insides of the fiber. And typically, we grow the cells on the outside of the fiber. What we have done then is set up a semi-permeable barrier between where the media is flowing and the cells are growing. It is a three-dimensional, more in vivo-like way of culturing cells that has a profound effect on how the cells behave in culture. This is just an illustration of what our hollow fiber cartridge looks like. We have a loop of silicone tubing where gas exchange takes place. We use a positive pressure displacement pump. There's a frictionless pumping system, so we don't shed plastic off the pump tubing and clog up the fibers. So a hollow fiber bioreactor, a three-dimensional capillary bioreactor, is fundamentally different from any other way of growing cells in three important ways. Number one, we have an extremely high surface area to volume ratio inside the cartridge, just like we do in vivo. So our standard cartridge is our C2011 cartridge. It has 4,000 square centimeters of surface area and a volume of 20 milliliters. So we basically have 200 square centimeters of surface area per milliliter volume. This extremely high surface area to volume ratio is the only way to culture cells at in vivo-like cell densities, 1 to 2 times 10 to the 8 cells per mil. It is the only way to culture cells at a density which allows them to control and generate their own specific microenvironment. This is super important. We'll talk about that later. Cells are bound to a porous support, not a non-porous 2D flask. This means that we never have to split the cells, and passage number is irrelevant. Nutrients come from the bottom up, just like they do in vivo. So it is a more in vivo-like way of growing cells. Again, we never have to split the cells. We've cultured a glioma cell line for over 2 years of continuous culture. We've produced a hybridoma for over a year of continuous culture with no change in antibody affinity and avidity. Lastly, we control the molecular weight cutoff. This is a measurement of the fiber pore size and the filter size. It is measured in Daltons. We have a 5,000 Dalton and a 20,000 Dalton molecular weight cutoff available, 5 kD and 20 kD. Now, I think it's very important to understand and know fundamentally exactly what a Dalton is. This is one of my pet peeves, and it will be part of the continuing medical education questions as part of this seminar, just to show you how important I think it is. And it's important to know that a Dalton is the mass of a hydrogen atom. It was first described by Sir John Dalton in 1784 when he was a professor at Cambridge University. So nowadays, NIST likes to call a Dalton 1/12 of the mass of a carbon atom. They want to relate everything to carbon. So when the aliens come, we can talk to them about carbon, and a Dalton is 1/12 the mass of a carbon atom. So a hydrogen atom is the smallest indivisible atomic unit that you can have. Just to keep things in perspective then, the antibody is a molecular weight of around 147,000 Daltons, 147 kD. The average mass of amino acid is around 120. So we can look at this and say, okay, IgG has about 1,200 amino acids. And it's important to understand this also in the context of an exosome. EVs are going to be anywhere from 60 to 120 nanometers in size, much larger than an antibody. So an antibody cannot cross the 20 kD molecular weight cutoff, and neither can an extracellular vesicle. So this is a cute little video. Our positive pressure displacement pump. It's a frictionless pumping system. Pulsatile flow facilitates exchange of nutrients and waste products across the fiber. Cells are growing on the outside of the fiber. Nutrients and waste products can cross the fiber. The product of interest cannot cross the fiber. We can simply harvest it by using some syringes and pulling out your product on a daily basis. So in the laboratory, the system is designed to be very simple. Temperature is controlled by the incubator. Gas composition is controlled by the incubator. It fits inside any standard incubator. We have a thin cord that goes through the door for power. It's not necessary to run anything through the back of the incubator, so we can maintain and retain our CO2 concentration in the incubator. When we work with the cartridge, it's very important to have good sterile technique. You must remember that Pasteur was correct. Life does not spontaneously generate. All we're going to do on a daily basis, move it into the hood, so we're in a laminar flow hood in a nice aseptic environment. Maintenance is only going to take 15 minutes a day. We want to measure the glucose, the rate of glucose uptake, i.e. the glucose uptake rate, or GUR, is going to be an indication of how many cells we have in the cartridge, how happy the cells are, and what's going on. So as long as the glucose uptake rate is either plateauing or increasing, the cells are very happy. We know from experience that a glucose uptake rate of approximately 1 gram per day means that we have about 1 times 10 to the 9th cells total in the cartridge. We're going to harvest our product. Harvest volumes will be anywhere from 2 milliliters on up to 60 milliliters. Concentration of our secreted products will be anywhere from 10 to 100 times higher than what you'll have in a regular bioreactor or flask culture. So the advantages of hollow fiber cell culture are as follows. We have a concentrated product. Product is concentrated by the molecular weight cutoff of the fiber, remains in the extracapillary space along with the cells, where it becomes very highly concentrated, and we can just harvest it out every day. We see uniform and complete post-translational modifications. This is because the cells are creating their own microenvironment, and are spending much more time in their optimum homeostatic physiologic cell culture conditions for optimum protein production and cell physiology. We see low apoptosis. The cells die, they're necrotic, but they don't split open. So it's not unusual to see large numbers of dead cells, intact dead cells in your harvested product, but we see significantly less contamination with intracellular DNA, host cell proteins, and most importantly, membrane fragments. We've developed our protein-free media, cGMP, chemically defined media for high-density culture. The cell culture environment found in a hollow fiber bioreactor is fundamentally different from any other way of growing cells. This makes it possible for us to specifically simplify and optimize the media that we use in conjunction with these bioreactors. So cGMP is chemically defined, protein-free, animal component-free, and cGMP compliant. One of the things that's important to me about cGMP is that it's not just a product, it is a direct manifestation of the different cell culture conditions found in a 3D hollow fiber bioreactor, because cGMP does not work in flask culture. cGMP does not work in standard cell culture systems. The cells must be at a higher density. They need to be generating their own microenvironment. So the cells need to be at least 5 × 10⁷ cells per mil or higher in order for cGMP to be able to support their growth. So we can see over long-term culture the consistency of production in terms of post-translation modifications, protein purification, and EV production over time. So, of course, we all know what exosomes are. We'll just do a brief review. They're small particles derived in biological fluids. They're anywhere from 30 to 120 nanometers in size. They have microRNA that may contain their therapeutic activity inside the vesicles. They have proteins on the exterior that are targeting proteins in my mind. My own personal theory and hypothesis is that EVs exist to protect the microRNA inside the vesicle, because you don't want to have free RNA floating around inside your biological fluids, and we need to be able to direct them to specific areas for them to exert their activity. So EVs are targeted cell regeneration vectors that can be used for many different therapeutic purposes. So just as a reminder, the standard methods for producing EVs, at least up until now, utilize 2D plastic flasks. We've already discussed how non-physiologic that is. And we'll take out serum. So get the cells to confluence in a serum-containing media. We remove the serum. Serum contains a significant amount of endogenous extracellular vesicles. We want to get rid of those and not have them interfere. So we'll put them into protein-free media, serum-free media for about 48 hours, and then collect the EVs under these conditions. So you can imagine just how non-physiologic, even in the absence of serum, that these kinds of conditions for EV production are. And just a little experiment that we did in-house, we found that if we use cGMP under these conditions, that we can significantly enhance EV production from cells in this protocol. However, it's still not very efficient, and keep in mind that 10⁹ cells generates a lot of plastic trash. And in this case, this is just an example of what it takes to expand a T75 flask up to 10⁹ cells, which is going to be about 50 T225 flasks. So it generates a lot of garbage. So there must be a better way of doing things. So the advantages of hollow fiber bioreactors for EV production are that large numbers of cells can be cultured in a small space. Our standard cartridge, the C2011, can support 10⁹ cells. Our larger cartridge, which we're going to be scaling up to in the next few weeks in our own lab, the C2018, can support 1 to 3 to 5 times 10 to the 10th cells. It's quite a range, I know, but somewhere between 1 and 5 times 10 to the 10th total cells in a volume of 60 mils. The secreted exosomes will be significantly concentrated. We're also finding that harvest frequency has a significant impact on the efficiency of extracellular vesicle collection. So we're looking to optimize that, and I'll give a little more data on that in a minute. So we know we can do continuous production of many cell types over many months. So we can set up a continuous production scheme with a 3D hollow fiber bioreactor. We have protocols where we've used serum to support the cells. We can put serum into the circulating media, through the ICS, or intracapillary space. But the factors in serum that support cell growth can cross the fibers. The extracellular vesicles, the endogenous extracellular vesicles that we don't want, cannot cross the fiber. So we can safely use serum in the, to support the cells if we need to. However, we found that most cell types, as long as we can get the density up to 10 to the 8 cells per mil, we can utilize cDMHD, which of course is going to be EV free and is fully cGMP compliant. MSCs, mesenchymal stem cells, we worked with a lot. I'm not going to go into be a whole another seminar to talk about MSCs, but in brief, we found that mesenchymal stem cells, regardless of their source, don't seem to proliferate too much unless they're under special conditions. I believe that this is just a function of MSCs and how they behave in vivo. Part of the definition of MSCs, mesenchymal stem cells, is that they attach to plastic and proliferate. In a 3D environment, they do not appear to proliferate. This is both good news and bad news. Bad news is we need a lot of cells to seed a cartridge. Good news is if the cells aren't proliferating, they're not differentiating. So I'll show that we've been able to maintain MSCs for 45 to 60 days, continuous culture, harvesting exosomes all along. Just some of our initial work showed that with the human adipose-derived mesenchymal stem cells, there are one C2011 bioreactor harvesting once a week. We were generating about, we'll just say 9 times 10 to the 11th, we'll fudge it up to 10 to the 12th EVs. And this equaled, in terms of our control, 540 T225 flasks. EV production is going to vary a lot in terms of our comparisons, depending on the cell type, depending on the harvesting frequency, and many other factors. But this was a direct comparison that we did one of our early ones. And in terms of bioactivity, we can demonstrate that we did see enhanced wound healing activity produced by these exosomes in this rat wound healing model. But more interestingly, as collaborators of ours that we worked with, this was a way to make extracellular vesicles inexpensive enough that they could be used in a commercial product. So in point of fact, we have this product produced called Exoface. There's an exosome-based facial moisturizer, and they've got some data on this, 95% reduction in wrinkles, skin luminescence, some 18% acceleration of cell renewal. So we've got lots of nice data from this commercial product that utilizes our system to produce commercially viable extracellular vesicles. In this case, they're called Smarticles. This is just another example from one of our researchers that's been working working with us from Switzerland. And basically what it's showing for our small cartridge, this is our C2025D, 450 square centimeters of surface area, that we get cumulative linear growth of the EVs and linear production of EVs over time. This is bone marrow MSC. From human umbilical cord MSC, in this particular case, I think we found perhaps, so that this is data that has been published, but there's still some data that I still need to get from them. But in this case, we can see that they seeded 2 times 10 to the 8th MSCs, and they utilized the cell growth factor that seems to have gotten the cells to proliferate. In any event, they seeded 2 times 10 to the 8 cells, and we consumed 200 mg of glucose per day, we expect, and we see that they over time, they did expand, and we were able to culture these MSCs for 44 days before things started to drop off, but this is going to be important in a minute. And basically, they compared 2D MSC extracellular vesicles in terms of cell surface markers with the 3D EVs and their cell surface markers. And here we're just showing consistency between day 23 and day 55 of culture, which to me is important because EVs remained uniform and consistent over time, even though the cell population started to diminish. So I think that this is showing that the physiology of the cells was still optimum for EV production. Here we've just got a comparison in terms of particle size and Western blot analysis of the 2D versus 3D EVs. In this case, we can see that the 3D EVs equaled about 225 T225 flasks. Now, our poster from International Society of Extracellular Vesicles, we did in conjunction with Laura Perrin. She had some human amniotic fluid stem cells that we cultured in our little cartridge and produced EVs for her. And most surprisingly, we found that regardless of the harvesting interval, whether it was 1 hour, 2 hours, or every 24 hours, EV concentration was pretty much the same in the level of around 2 times 10 to the 8th, 4 times 10 to the 2.8 times 10 to the 10th, 4 times 10 to the 10th, the hourly harvest. Or if we harvest it over a day, concentration is the same. So there's a significant either inhibition of EV secretion based on external EV concentration, or inhibition of secretion based on this concentration. So it's either inhibition of reuptake or inhibition of secretion based on the high concentration of EVs in the extracapillary space. Here's some of our primary data, just showing the same particle size distribution, the EV numbers. Tetraspanin analysis showed weaker expression of some of the other CV markers. We're not exactly sure what's the reason for that, because the bioactivity was actually enhanced in terms of both fibronectin co-precipitation and in vivo injection to Alport mice, showing amelioration of proteinuria based upon the EV therapy of the kidneys. So the advantages here, we got demonstrated enhanced bioactivity in many different publications. We mimic the mammalian circulatory system, long-term homeostasis of cell culture environment. Now, the most important thing to me is going to be that many grant applications are being criticized because there's just not the capability and capacity to produce the number of EVs required for clinical trials or in vivo animal studies. My feeling is that hollow fiber does actually produce the numbers of EVs required for clinical translation. And we have a project ongoing in our lab that we're just starting up, where we're going to try to demonstrate the production of 10 to the 15th or gram quantities of EVs. And if we look at other comparative methods that I pulled out of some journal articles and some presentations, Rooster Bio is promoting a microcarrier method for producing large numbers of EVs, 10 to the 13th EVs. So that would actually require, to get to 10 to the 15th, would take 100 runs of this bioreactor to produce 10 to the 15th EVs. Quantum has published some data showing that they get 10 to the 13th in 1,500 mils of supernatant. And we need to, again, run 100 runs of their system in order to get to 10 to the 15th. My expectation is that we'll set up a cartridge and be able to generate 10 to the 14th EVs in our product for under, in a month, for under $30,000 of cost. What would a system like this look like? Just to look into the future, maybe a little bit. This is a diagram of a closed, automated, cGMP compliant hollow fiber bioreactor system that could be used to produce 10 to the 14th, 10 to the 15th EVs or more. So we can see that the hollow fiber bioreactor is the most evolved way, the most in vivo-like way of culturing cells. And I see great applications for this, both in terms of producing EVs under physiologic conditions and producing EVs in quantities sufficient for clinical translation. So again, I'm John Cadwell with Fiber Cell Systems. Thanks so very much for your time.